With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hi everyone, our question says in a polymerase chain reaction, after the denaturation step, why the mixture needs to cool down to a lower temperature. In this question, we have been asked that in PCR, why the reaction mixture needs to be cooled down after the denaturation step. So PCR is a technique which is used to amplify certain region of DNA, and this PCR consists of 3 steps. The first step in PCR is denaturation, in which double-stranded DNA is converted into single-stranded DNA. So here double-stranded DNA gets converted into single-stranded DNA, and for this purpose we require very high temperature to denature the DNA. So high temperature is required, which is about 94 degrees Celsius. And the second step after denaturation is annealing. So in this step we utilize primers, and the primers anneal to the single-stranded DNA. So we utilize primers, and these primers are complementary sequences which anneal to the single-stranded DNA. Anneal to single-stranded DNA. And for this process we require a low temperature range. So we require low temperature for this purpose, and the temperature should be between 40 to 60 degrees Celsius. So as low temperature is required so that the primers can anneal to the DNA, that's why we need to cool down the reaction mixture. So here the correct option would be option A, that is to permit specific annealing of the primers. Option B, to give halt to the reaction mixture, is wrong. We do not need to give halt to the reaction mixture. C, to increase the activity of the enzyme Taq polymerase, is wrong because Taq polymerase is utilized in the last step, that is extension step. So there is no use of cooling down so that the Taq polymerase enzyme activity can increase. So this is also wrong. D, to obtain multiple copies of DNA. So this is also wrong because multiple copies of DNA will be obtained after following all these 3 steps. So we do not need to cool down mixture to get multiple copies of DNA. So the correct answer for this question would be option A, to permit specific annealing of the primers. So this is the answer. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.